The last video dealt with an idea of how the very earliest steps of life on Earth happened, turning inorganic molecules into organic molecules. This video outlines how scientists think the next steps happened. This is video 3.4 on the origin of cells in endosymbiosis. This video will cover these two objectives. Press pause to take a closer look. One thing I want you to remember is that the origin of life didn't happen in just one step, and it wasn't a quick process. The origin of life had 700 million years to take place. Now, the story I'm going to tell is to take the spontaneous abiotic origin theory to its completion. So, the Miller and Urey experiment showed how organic monomers could have arose in early Earth conditions. Next, those monomers had to combine into polymers. The polymers had to assemble into cells, and then those cells had to go from simple prokaryotic cells to more complex by evolution. In their experiment, Miller and Urey found amino acids. Amino acid monomers are the building blocks for protein polymers. The other macromolecules essential for life are lipids, carbohydrates, and nucleotides. If amino acids could have formed under early Earth conditions, then why not the other macromolecules? Nucleotides are the building blocks for DNA and RNA, and there's growing evidence that RNA was the first genetic material and not DNA. If you think about it, the origin of genetic material was huge. Genetic material makes inheritance possible, and inheritance makes evolution possible. So instead of each organism having to restart after each generation, RNA allowed for successful traits to be saved and act as a starting point for the next generation. Next, those macromolecule polymers are thought to have self-assembled into primitive cells called protobionts. Protobionts are basically a membrane with protein inside. Having a membrane bubble allows processes to happen on the inside of the bubble that are different than the processes that happen outside of the bubble. Later, protobionts are thought to have evolved into the first, simplest prokaryotic cells. Finally, prokaryotic cells gained complexity to make them eukaryotic cells. This step is going to be a main focus of the video, so I'll save the details until later. The rest of this video will be dedicated to the origin and evolution of cells. Remember, the processes that lead to making cells as we know it took a very long time with a lot of steps. One hypothesis about how this happened is that the ocean became kind of a soup of macromolecules, a mixture of all of those molecules essential for life. Because of biochemical properties that we'll talk about in a couple of units from now, lipids like to be next to each other, especially when the lipids are in water. Keep in mind that lipids are the main components of membranes. So scientists think that lipids self-assembled into these membrane bubbles. In the process of making the bubbles contained some of the other biopolymers like RNA, proteins, other lipids, and carbohydrates. And once these bubbles started to function differently because of their contents, we can call the bubbles protobionts. Protobionts are the precursors for cells. There are a couple of important things going on. First, the membrane of protobionts separates the inside from the outside. Why is this important? Well, the inside can become an environment that's favorable for metabolism chemical reactions, even if the outside is not as favorable. Second, the protobiont can contain its own genetic material. When protobionts self-replicate, another huge step, the protobionts can pass on genetic material from one cell to the next, essentially saving all of the favorable traits and creating a pathway for evolution. Scientists have done a proof of principle experiment to show that protobionts are possible. Creating a mixture of biopolymers in a test tube, scientists found that membranes do spontaneously form these bubbles. Not only that, but they found that these lipid bubbles can undergo metabolic chemical reactions, shown over here, and even reproduce, like you can see over here. This is pretty wild. So, does this prove that cells came about this way? Well, no, but the experiment shows that the idea is feasible. So if we accept that protobionts were early cells, then protobionts were the starting material for the most simple cells, prokaryotes. This is where having genetic material is huge. With their genetic material, protobionts could have undergone mutations that make them more complex until eventually cells ended up as prokaryotes. Now, I've been throwing around this term prokaryote like you should know what it means, and I just want to make sure. The two major types of cells we'll talk about in this class are prokaryotes and eukaryotes, and here's a whole list of things that compare the two. But these lists can boil down to these simple points. Think of prokaryotes as simple cells with no nucleus, and eukaryotes as more complex cells with a nucleus. One reason that prokaryotes are simple is because they don't have any membrane-bound organelles. Eukaryotes do have membrane-bound organelles like endoplasmic reticulum, Golgi apparatus, vesicles, vacuoles, and more. Finally, it's good to have an example of each. A good example of prokaryotes is bacteria, but you can be even more fancy by giving a specific type of bacteria like E. coli or Staphylococcus. Eukaryotic examples include plant cells, animal cells, yeast, and protozoa, and if you're looking for a fancier example, think neurons, sperm cells, amoebas, or paramecia. So, how did nature bridge the gap between prokaryotes and eukaryotes? Well, one idea is called the Infolding theory. Remember, a big difference between prokaryotes and eukaryotes is that eukaryotes have membrane-bound organelles. Well, Infolding theory explains the origin of these organelles. The idea is that the membrane of prokaryotes infolded and broke off, and when the membrane pieces broke off, that created the early versions of organelles, things like the nucleus and endoplasmic reticulum. The good thing about having organelles is that each organelle creates a specialized compartment with a microenvironment specialized for a certain job. After a while, it became advantageous to have these infolded structures, and cells with infolding became modern-day eukaryotes. There's another theory out there that explains how eukaryotes got chloroplasts and mitochondria. Chloroplasts and mitochondria arose by a mechanism different than infolding. They arose by endosymbiosis. The endosymbiotic theory outlines the origin story of mitochondria and chloroplasts. So, what is the endosymbiotic theory? Well, the theory says that early eukaryotic cells engulfed specialized bacteria. The bacteria gained the advantage of being protected from the environment by living inside of this big eukaryotic cell. The eukaryotic cell benefited by the specialized job of the bacteria helping out. Aerobic bacteria are really good at making ATP. In fact, aerobic bacteria can make ATP 18 times better than anaerobic bacteria, or anaerobic organisms in general. Can you imagine being able to do something 18 times faster? It's thought that these aerobic bacteria eventually became mitochondria, the ATP-making specialized organelle of eukaryotic cells. Photosynthetic bacteria can make food, glucose, from 3 common ingredients: carbon dioxide, water, and light. For a cell that couldn't do photosynthesis, engulfing a photosynthetic bacteria would be like having your own chef live in your house. And it's thought that these photosynthetic bacteria eventually became chloroplasts, the photosynthesis masters of eukaryotic cells. Let's check out these ideas more closely. Here's the origin story of mitochondria. At first, you see a simple eukaryotic cell. Now, that early eukaryote finds an aerobic bacteria cell and engulfs the cell by a process called phagocytosis. Now, normally, phagocytosis is how single-celled organisms get food, but this early cell didn't digest the aerobic bacteria. Instead, the aerobic bacteria lived inside of a vesicle in the eukaryote. The bacteria gained protection from environmental hazards by living in the eukaryote, and the eukaryote gained the ability to make ATP 18 times more efficiently than it would be able to on its own. Now let's see how chloroplasts arose. Here's a eukaryotic cell that has mitochondria. Now, that cell is engulfing a photosynthetic bacteria cell, but not digesting the engulfed cell. Again, the engulfed bacteria benefits by living inside of this big old eukaryotic cell and being shielded from environmental hazards. The eukaryote benefits by being able to make its own food, glucose. Now, every cell doesn't get its mitochondria and chloroplasts this way. That's just how it happened first. Eventually, the relationship between the eukaryote and the engulfed bacteria evolved so that they couldn't be separated. And at that point, we'd consider the engulfed bacteria, mitochondria, and chloroplasts. A fancy name for this relationship is called obligate symbiosis. And what this means is that at some point, if you took mitochondria or chloroplasts out of the eukaryote, it wouldn't be able to live on its own like the original bacteria would have. Endosymbiosis is such a cool theory for a bunch of reasons. First, endosymbiosis is a really cool synthesis of a lot of different types of undeniable evidence. And second, one thing that's unique to this class so far is that endosymbiosis is an idea originated by a female scientist, and we haven't had any example of female scientist pioneers in the class so far this year. And this is a color photo, meaning that this discovery happened relatively recently by Lynn Margulis, who has University of Chicago connections. Now, when you think about the evidence for endosymbiosis, it may be easier to categorize the evidence into structural evidence, genetic evidence, and functional evidence. Let's examine each in more detail. When you consider the evidence for endosymbiotic theory, this is what I expect you to be able to do. First, describe each specific piece of evidence, and second, explain how the evidence supports the endosymbiotic theory. Well, let's take a look at the structural evidence. Basically, mitochondria and chloroplasts look like bacteria. What do I mean by that? Well, mitochondria and chloroplasts, abbreviated as M and C on some of these slides, have a similar size and shape as bacteria. Now, I know that's not too strong of evidence, but there's more. Mitochondria and chloroplasts have internal structures that independently living bacteria have. These structures include having their own DNA and their own ribosomes, which aren't shown on this picture. What's more than this is that the structure of the DNA in mitochondria and chloroplasts looks like bacteria DNA. The DNA of mitochondria and chloroplasts is in a single circular chromosome, just like bacteria. And this is unlike eukaryotic cell DNA, which is in several linear chromosomes, not a single circular chromosome. So, how does this support the endosymbiotic theory? Well, we know that similar structures can be indicators of evolutionary relatedness. The fact that mitochondria and chloroplasts have a similar size, shape, ribosomes, and uniquely shaped DNA, same as bacteria, suggests that mitochondria and chloroplasts are related to bacteria. I want to expand on the DNA of mitochondria and chloroplasts as genetic evidence in support of the endosymbiotic theory. And keep in mind that the eukaryotic cell as a whole has its own DNA. Mitochondria and chloroplasts have their own separate DNA. The DNA of mitochondria has genes and that encode proteins essential for aerobic cellular respiration, and the DNA for chloroplasts has genes that encode essential proteins for photosynthesis. And here's how mitochondria and chloroplasts having DNA supports the endosymbiotic theory. Now, having DNA and ribosomes is a characteristic of all living things. So having separate DNA suggests that mitochondria and chloroplasts were once separate living things. Another weird thing about mitochondria and chloroplasts is that both reproduce independently from the cell. You probably know from your previous biology class that eukaryotic cells reproduce by mitosis. Well, mitochondria and chloroplasts can reproduce themselves even when mitosis is not happening, as shown right here. And the process by which mitochondria and chloroplasts reproduce looks very different than mitosis. In fact, mitochondria and chloroplasts reproduce by a process that looks like binary fission. Binary fission is the process used by bacteria to reproduce. So, how does this fact that bacteria, mitochondria, and chloroplasts reproduce similarly support endosymbiotic theory? Well, the fact that they reproduce on their own, a characteristic of living things, and the fact that that process looks like how bacteria reproduce suggests that chloroplasts and mitochondria are related to and were derived from separate living bacteria. Let's zoom out and see this origin of life story as a whole. According to spontaneous abiotic origin theory, conditions on early Earth supported the creation of organic monomers from the inorganic chemicals that were present at the time. And this was shown to be possible by the Miller and Urey experiment. Once organic monomers were in place, the monomers had to self-assemble into macromolecule polymers, and then the polymers had to assemble into primitive cells. Going from polymers to cells happens in multiple steps. First, the very primitive cells called protobionts were formed. When protobionts had genetic material and could reproduce, this gave protobionts the ability to evolve into prokaryotic cells. Then, prokaryotic cells evolved into more complex eukaryotic cells, again through multiple steps. First, the membrane of prokaryotes folded inwards to create internal compartments called organelles, and then eukaryotes gained chloroplasts and mitochondria by endosymbiosis. Here's a graphic that summarizes the part after prokaryotes are formed. So, at the earliest time, we have a prokaryotic cell. The prokaryotic cell became an early eukaryotic cell when the plasma membrane folded in on itself, creating organisms like the endoplasmic reticulum and the nucleus. Next, the early eukaryotic cell engulfed an aerobic bacteria. This gave the eukaryote the ability to make ATP 18 times more efficiently than it could, and gave the prokaryote a protective home. Over many generations, the bacteria evolved into a mitochondria. Next, some of these eukaryotes with mitochondria engulfed photosynthetic bacteria. This gave the eukaryote the ability to make its own glucose by photosynthesis and gave the bacteria a protective home. Over many generations, the bacteria evolved into a chloroplast, and these cells further evolved into the cells that we have today. I think the last two slides did the job of recapping what was covered. If you'd like another recap, pause the video and read through this.